Well, I get to make the next presentation. I'm going to really focus on one organism and actually one molecule of one organism. So it's really an extremely reductionist approach, but it may exemplify what microbes in the intestine can do. I think SFB is certainly extremely important in that regard. And the work I'll tell you about balances that according to the type of scale that Nadine had drawn. The organism that I'm going to talk about is Bacteries fragilis. It's a strict obligate anaerobe that you can grow. It's non-spore forming. It's common in the normal flora of many mammals, particularly humans. This organism's genome was sequenced about 10 years ago, and what was found was that remarkably for any microbe that we knew about, but certainly for microbes that are involved in the human at that time, this organism had loci for the production of 8 different polysaccharides. As you can see on this genome map, most organisms that we know about make a single capsular polysaccharide, yet this organism had 8. It appears that this motif now of multiple polysaccharide production, multiple capsular polysaccharide production, is actually quite common in the Bacteroides of the human gut. Remember, one gene does not make a polysaccharide. These are loci with 8 to 25 genes in them, and we very uncleverly, I would say, name these polysaccharide A, B, C, D, E, F, G, and H. Polysaccharide A was the most abundant of these polysaccharides, present on about 60% of organisms. Now I'm talking about from a single colony pick in a broth culture. 60% of the organisms express polysaccharide A. Others express other polysaccharides. Occasionally an organism can express 2 or 3 polysaccharides. So we focused on polysaccharide A. This is the structure of polysaccharide A. There are 4 sugars in the repeating unit of polysaccharide A. They're all derivatives of galactose. Remember, this is a polysaccharide, so this one repeating unit of 4 sugars is glycosidically linked from this end to this end about 100 to 200 times in each molecule. So it's much larger than it appears to be on the board. Another striking characteristic of this polysaccharide was that it had a free amino group on this sugar. When bacteria, and in fact when eukaryotic cells make amino sugars, we N-acetylate them, like here and here. But this organism actually synthesized this polysaccharide with a free amino group here. It also had a negatively charged carboxylate group on this pyruvate ring of this galactose here. So this positive and negative charge led us to call these polysaccharides, which are very few, I would say, of the hundreds and hundreds of polysaccharides which have now been structured. There are about 5 or 6 that have this zwitterionic motif, positive and negative charge all the way up and down the chain. I think we all learned in immunology that polysaccharides were T-cell independent antigens. They cross-link B-cell receptors. They cause the B-cell to make IgM antibody, but they don't elicit help from a T-cell. So therefore, these B-cells don't undergo immunoglobulin switching, and they don't undergo B-cell maturation. What was interesting about polysaccharide A is it actually stimulated T cells, and it specifically stimulates CD4 T cells, And these are, I think everyone's familiar with the various phenotypic markers of CD4 T cells and functional characteristics of the CD4 T cells. But we're going to talk about one today, although it also affects interferon gamma production by Th1 cells. I'm going to talk about one function today, and that is by FOXP3 IL-10 producing regulatory T cells. So the first thing is, how does this antigen stimulate T cells? Well, this is an antigen-presenting cell. Let's call this a dendritic cell. It is taken up into a vesicle, an endocytic vesicle. It is depolymerized to a smaller size. It is not, as proteins are broken down from proteins to peptides by proteases. In fact, there aren't glycosidases that do that in the endosome. This is an oxidative mechanism. On PSA, it happens to be a deamination reaction induced by nitric oxide. On other polysaccharides, there are reactive oxygen species in the endosome that get upregulated and depolymerize them. And then this molecule goes through the regular complex MHC2 pathway, ultimately with this oligosaccharide replacing the self-peptide and getting presented to a T cell. But it's the nitric oxide that's different. Why aren't other polysaccharides T cell inducing? Well, it has to do with that charge motif I told you about. So I've color-coded this so that negatively charged polysaccharides are red, blue are the positively charged, and the zwitterionic are positively and negatively charged. They all get into the endosome. They all get broken down by these various oxidative reactions I talked about. But the only ones that seem to replace the self-peptide in the MHC2 molecule are these zwitterionic polysaccharides. The others don't do that. These replace the self-peptide, and they actually bind to MHC2 in a way that conformationally we don't understand yet. But it certainly is electrostatic due to the positive and negative charge, because if we modify the positive charge chemically or the negative charge chemically, in fact, they no longer do this and they're no longer presented to T cells. But it's the zwitterionic motif that's essential for their binding to MHC2 and T cell presentation. And this is just a confocal picture of PSA in red in the endosomes of this. This is actually a B cell. And the MHC2 is in green. The alpha beta T cell receptor is in blue, and this white area of trilocalization is PSA being presented to the T cell. Now, what's required for IL-10 induction? Because I told you that's what I was going to talk about. Well, if you make, I'm looking at IL-10 production on the y-axis in both of these graphs. If you look on the left, we looked at various combinations, dendritic cells with T cells. We stimulated them with anti-CD3, a common thing that immunologists do. The important point here is if you use T cells without dendritic cells, you get very little production of IL-10 by PSA. However, if you use dendritic cells with T cells and anti-CD3 in this case to kind of goose up the T cell, you get lots of IL-10 being produced. And that this IL-10 is being made by the T cell and not the APC is shown here because, again, here's the wild-type T cell and the wild-type CD4 cell, lots of IL-10, but if you use an IL-10 deficient CD4 cell, but the same dendritic cell, in fact, there's no IL-10 being produced. So the T cell in this reaction is made by, makes the IL-10. The other interesting thing about TLR2 is it's also a TLR2 agonist. This just shows HEK cells transfected with TLR2 or TLR4, and you can see PSA stimulates. This was IL-8 was the readout here. Lots of IL-8 on the HEK infected TLR2 transfected cells. This is just a control known stimulant of TLR2, and there's no TLR4 effect. And it turns out that the TLR2 in the dendritic cell is important for IL-10 production by the T cell. So again, I'm looking at IL-10 in a co-culture, wild-type dendritic cells and wild-type CD4 cells, lots of IL-10 being produced. If you use a TLR2 deficient dendritic cell with a wild-type T cell, about a third as much IL-10 is being produced as with the wild-type cells. So TLR2 is quite important in IL-10 production, although there is some induction of IL-10 beyond that. And then to take this to a model, this is actually the model that Fiona Powry has championed. And what we did was we fed mice PSA over the course of this experiment. These were RAG-deficient mice that had been given a Helicobacter that is a probiont, if you will, in this situation. By itself, it doesn't cause disease, but if you give these RAG mice CD45RB high cells, effector T cells that are presumably naive, in fact, they get lots of colitis, as you can see here. This is a blow-up. This top row is all at the same magnification. But if you PSA treat them, in fact, they have a markedly reduced level of colitis, as you can see. Again, this is a blow-up, lots of inflammation here, and essentially none to little here. So is this IL-10 mediated? Well, in this experiment, this was just a co-culture experiment with the Helicobacter and with PSA, and then with an antibody to the IL-10 receptor. This is different MOIs of Helicobacter, and you can see a dose-response effect. This is TNF-alpha, the pro-inflammatory cytokine is the readout. If you put PSA into this DC T-cell co-culture, in fact, a marked reduction in TNF-alpha production. But if you also add an antibody to the IL-10 receptor, lots of TNF-alpha being produced. So PSA seems to have an anti-inflammatory effect mediated by IL-10. And then going back to this model, in fact, we did treat animals with the anti-IL-10 receptor antibody. The controls were the PBS treated in this complete model with Helicobacter and CD45RB high cells. PSA treatment, again, protected, but if you treated the mice with an antibody to the IL-10 receptor, the protection mediated by PSA was abrogated. Was this IL-10 being made by T cells? We did the same model, but this time the CD45RB high cells came from IL-10 deficient animals. Remember, you're putting them into RAGs, so they have no T cells of their own. And if you just look at the dots, not the colored, in fact, PSA was no longer protective. These are weights of animals, the colors. So low weight is good. I'm sorry, low weight is bad, the high weight is good. So PSA does seem to mediate its effect in this experimental model by induction of IL-10 in T cells, CD4 T cells specifically. And in another model of colitis, I got tired of 2-month models, so we switched to TNBS, which is a chemical model of colitis, presumably mediated primarily by the adaptive immune system, and this has been shown by several groups. And PSA also protected in this model. This is clinical score here, and the protective effect is here, but not in mice that were TLR2 knockouts. There was no longer a protective effect of PSA. So the in vivo and the in vitro data seem to correlate with each other quite well. I'd like to tell you a little bit about one other model of that we've been looking at, and that is the EAE model. This, as you, I think, know, is a mouse model that represents multiple sclerosis. It is not multiple sclerosis any more than experimental IBD is Crohn's disease, but it does have many immunologic aspects that are similar. What we did, in this model, you give the animals a peptide from myelin basic protein, along with complete Freund's adjuvant. You give it on day 0 and day 6, and then this is neurologic symptoms on the y-axis, and all the way up here are animals that can't walk. They're so neurologically debilitated. And you can see in the PSA, so PSA was started here and given every 3 days through the course of this slide. And you can see the marked protective effect in terms of neurologic symptoms in the EAE model. And this is the histology of that. PBS in the left column, purified PSA treated in the right. There's demyelination here, and there isn't here. And there are nucleated cells, which my neurology colleague tells me are typical in the brain of patients with EAE, with multiple sclerosis, and you see few in the purified PSA treated group. Interestingly, what we did was we started PSA at several different points in time after the initiation of the disease. So again, to initiate the disease, you give the myelin basic protein on 0 and 6 days. So we started PSA on either day 3, 7, 10, or 16. The PSA treatment ameliorated the course of disease whenever it was started. It didn't reverse it. It just stopped the progression of the disease, which presumably still is ongoing until it was given by various immunologically mediated phenomena. Is this TLR2 dependent? Yeah, it is. This is an experiment now with TLR2 knockouts. The protective effect of PSA in the controls is not seen, as I think is pretty obvious here. So this is TLR2 dependent in this mouse model of EAE, and it's also IL-10 dependent. If you use an IL-10 knockout mouse, you no longer see protection from PSA. So the same theme is through all the things I've told you. Protection requires IL-10 and TLR2. Just to summarize then what I've told you today, this is a dendritic cell. This is a naive CD4 T cell. PSA is from the organism is taken up by the APC. It's processed by a nitric oxide dependent mechanism to a smaller molecular size. PSA is also an agonist for TLR2. I will add that there are other signaling pathways involved that we're exploring now, but TLR2 is obviously critical from the data I showed you. What this does, at least, the TLR2 signaling, is it makes more nitric oxide in the endosome, which is required for PSA degradation. It makes more CD86, B7-2, which is required for signaling to the T cell, and it induces more MHC2, which is obviously where PSA is loaded for presentation. So it's a neat example of the adaptive and the innate immune system working together to stimulate this naive cell to become a FOXP3 regulatory T cell, which makes IL-10 and then protects in the EAE model, the colon in the IBD model. And we've looked at another model of post-surgical adhesions, and it's also protective in that model. All three by the same mechanism that I talked about. Who did the work? The colitis work was done by Sarkis Mazmanian when he was in my lab, and then continued in his lab at Caltech, along with a postdoc of his, June Round. The work on the dendritic cells been done by Surya, the TLR signaling by Chun Wang, Brian Cobb, Jinyu have done the antigen presentation work. My brother actually happens to be a neurologist, so it was very convenient to learn the EAE model and get it done. He does it in his lab, and his postdoc, Javier, they did most of that, and then Laurie Comstock has done most of the genetic work that I talked about. Thank you very much.